processes. Glomerular filtration, all right?
Tubular reabsorption.
Wait. Is there any hormone that directly regulates glomerular filtration rate? We have hormones that bind. Renin's not a hormone. It's an enzyme.
That's a sodium.
So, pardon? We'll talk about it in a little bit. So is there any hormone that binds to the cells of the podocytes or to the glomerular capillaries that changes filtration rate? Pardon? Angiotensin is, that affects systemic, and it's activated at the glomerulus, all right, but its effects are systemic. So we have the macula densa releasing prostaglandins, and that's about as close as we get to hormones, okay? Are the prostaglandins released by the macula densa during autoregulation? They're not traveling in the blood, so they're more like a paracrine effect than a hormone chemical that's transported in the blood. But we don't have any major endocrine organ that's going to be releasing hormones. Okay. So what was the next one?
Tubular reabsorption.
So tubular reabsorption, and we talked about this occurring in the proximal convoluted tubule. Today we'll look at additional sites. And so this again is directed from filtrate to blood, peritubular blood. Is this affected by hormonal control at the proximal convoluted tubule? No. The only reabsorption that is affected is when we look at electrolyte balance. I'll put this in parentheses. Parathyroid hormone decreases reabsorption of phosphate. But primarily our hormonal control is going to be at the distal tubule. Okay? And then the third step, tubular secretion, which is blood filtering. Now, when we have molecules that are being filtered, we end up with the same concentration in the efferent arteriole as we do in the nephron. Okay? So our 300 milliosmoles, we still have 300 milliosmoles. We have the same amount of glucose in the filtrate as we do in the blood. Okay? Because it's moving across on equal. I had trouble with that one this morning. It's equilibrated. It's moving across in an equal flow rate. And part of the reason we then see continued movement, if it's still equal, is because the blood pressure is so low, the hydrostatic pressure is so low in our peritubular capillaries. So it makes it easier for material to be taken up from the proximal and distal convoluted tubules. So now we have our proximal convoluted tubule. Get a little visual here. And what have we? We've done reabsorption. So what kinds of substances have we moved out of the proximal convoluted tubule? Glucose. So by the time we get to our nephron loop, all of the glucose is gone. Okay, certainly water and sodium ions, about 65 to 80% of that has been reabsorbed from our proximal convoluted tubule. Okay. Other ions, amino acids, all. And then a variety of ions, phosphate and magnesium ions and a bunch of other stuff. So of the material that we have done, well, let's finish it with reabsorption. I'm sorry, let's finish with secretion. So from the peritubular blood, what are we secreting? One of the major components, we'll look at it tomorrow, is hydrogen ions. Okay? So we're going to secrete hydrogen ions, and we're going to reabsorb bicarbonate ions. And that's one of the major mechanisms by which the body adjusts an acidic pH. Another thing that we, do you remember what I talked about? Another thing that we secrete. You would think that because of the reabsorption of sodium, remember we had a sodium-potassium pump, and so we take out sodium, potassium goes into the cell. However, most of that sodium actually leaks back out again between the cells. All right, so we really don't see an increase of potassium in our filtrate. But we do have the drug breakdown products, so the metabolic products of drug breakdown, drugs and other components that are protein substances that are bound to plasma proteins. So plasma protein bound components. These are secreted. Would they be filtered? If I have a hormone that's bound, that's how we transport thyroid hormone, remember, is bound to a plasma protein. Would I secrete that into my filtrate?
No.
Proteins are too large to cross between the slits or through the basement membrane, and so they would not be in the filtrate, but we can see them as from the glomerulus, but we can end up seeing them in our urine because of secretion. Okay? So this is how many drug products get into the urine. They're too large to go through the filtration, but they're secreted at the proximal convoluted tubule, and they're added to the filtrate from that position. All right, so now we're ready to move into our nephron loop. And this would be our descending limb and our ascending limb of the nephron loop. And looking at the slide on the screen, the nephron loop functions to provide what? Sorry? Does it function to concentrate the filtrate? To dilute the filtrate? Or to form the concentration gradient? It's both B and C, because it ends up diluting the filtrate in the process of forming the concentration gradient. Okay? That's really not its function, that just is that's how it does it. So, a couple of characteristics to think about. These are listed for you on page 24, top of page 24 in your lecture notes. And I'm going to kind of describe this, use some verbiage before I show you the images on the screen. Our blood has a content, a range of particle concentration, which is 300 milliosmoles. That 300, that number, 300 milliosmoles, is pretty standard for blood and for interstitial fluid. So within the blood, it's going to be 300 milliosmoles. Outside of the blood, it's going to be 300 milliosmoles. Because of that equilibration I talked about inside the filtrate, it's going to be 300 milliosmoles. However, by the time this filtrate gets to our distal tubule, 100 milliosmoles. Okay? So coming into our nephron loop, it's 300, but when it leaves, it's 100. Obviously, we didn't concentrate the filtrate. We diluted it. Yes?
It concentrates on its way down and then dilutes on its way
Yeah.
back up.
So, where did those particles end up? If we have fewer particles in our filtrate leaving the nephron loop than came in, what happened to them? We move them out, not necessarily into the blood, but into the interstitial fluid. Okay? So as those particles are moved out, they end up creating ions, sodium and chloride, some urea ions, that are more and more concentrated the closer we get to the minor calyx. And so what we see is a concentration gradient that is similar, same as interstitial fluid in the cortex, and then becomes more and more concentrated as we move down to the papillary ducts. This same fluid surrounds the collecting ducts. And it is within the collecting ducts that we actually pull, it's from the collecting ducts that we actually pull fluid out and end up forming or not forming a concentrated filtrate. Yes?
I'm just over here, we have 300 milliosmoles.
Mm-hmm.
And then the interstitial, we have 300 because it equalized.
Mm-hmm.
But over there, we have 100. Is it still 300 in the interstitial over there?
Which means that there are some water channels that may or may not be present. We create this gradient because of a characteristic that occurs because this tube is carrying a flow downward. And, for a moment, this flow is upward. And what did we call that characteristic when we were talking about the testicular arteries and the pampiniform plexus? Counter current, we were transferring heat from the artery to the vein. And so we had a counter current exchange. Right? The flows of the blood were opposing each other, and thus we always had a difference in the amount of heat between the two, and we had a transfer of heat from one vessel from the artery to the vein. Even as the vein was getting warmer, the arterial blood was even hotter because it was closer to the abdomen. And as the arterial blood was getting cooler, the venous blood was already colder because it was coming from the scrotal sac. So we continue to have a counter-current exchange. So this is definitely counter-current. The flows are in opposite directions. There really isn't an exchange. We're not moving fluid from one tubule to the next, nor are we moving ions from one tubule to the next, all right? But there is counter-current flow. The ascending limb of the nephron tubule has sodium-potassium pumps and a chloride exchange, and so, but it is not permeable to water. Okay, that's one of the key characteristics. Is our ascending limb of the nephron loop does not have aquaporins. Every other structure from the collecting duct to the glomerular to the proximal convoluted tubule will have aquaporins, or they can be inserted under the influence of ADH, as in the collecting duct and the distal tubule. Okay? So without aquaporins, that means water cannot, significant amount of water cannot move out of the filtrate. So the ascending limb of the nephron loop is thus impermeable sodium, and chloride follows. There's also some urea, but primarily we look at sodium. So if we can't lose water and we're moving sodium and chloride ions out, then what's going to happen to the concentration of our fluid? It's going to go down, as we can see that it does. All right? So as we add these particles, and water can't follow. If water followed, then we'd have less volume, but we'd have the same concentration. Does that make sense? Okay? So if I have a pot, and it has a gallon of water and 2 cups of salt, and I only take the salt out, I'm going to have a gallon of water, but not as, this won't be as salty. Okay, whereas if I take a cup of everything, I have less volume, but I've lost both salt and water. So in this case, we're just taking the salt out. We still have the water, so our fluid becomes more dilute. Now, we've added extra sodium and chloride ions to this area, so the descending limb is permeable to water. So if I have a strawberry and I sprinkle sugar on it, some water is going to come out. What happens if I sprinkle even more sugar on it? Even more water is going to come out of the strawberry cells. So the effect of these extra particles in our interstitial or extracellular fluid is going to draw water out of our descending limb. What's that going to do to the filtrate in my descending limb? It's going to make it more concentrated. Okay? Now, the pumps that I have present here, the ATP requiring active transport pumps that I have present here, can only maintain a maximum difference of 200 milliosmoles. So if this is 300 coming in, and I put 300 here, and I pump out 100 milliosmoles of particles, that would make this 200, and make this 400. I can write those numbers up here. So if I have 300 and 300, I'm getting tired of red. So if I have 300 and 300.
But wouldn't that be higher than 300 at that point? Because it's concentrating down.
Hold on. And I move out 100 molecules, milliosmoles worth. This was 300 too, so this would now make it 400. Now this is higher than this side, so it's going to have an osmotic pulling effect because this is permeable to water. So this will now become 400. That's the maximum difference that I can create. I cannot make this 100 and that 500.
So the salts are going in there too? Oh no, the water's going in.
So that turned to 400 because it was drawing water out. So when the 400 in the interstitial drop back down?
Excellent point. In 30 seconds, I'll get to it. Okay? So now, if all we had was this counter current flow, that's the maximum difference that we could make. It would all be the same. It'd be 400 millisosmolality. That's the maximum we could concentrate our filtrate. Okay, it would be that 200 milliosmol difference. Instead, what happens, instead what happens is we get our filtrate as high as 1,200 to 1,400 milliosmoles. The surrounding fluid gets that high as well. And the maximum that we can concentrate our filtrate in the collecting duct is that number as well. Before I get to your question, let me answer your statement. Remember a particular series of blood vessels that branched off of the efferent arteriole? The vasa recta. Now this is going in this direction, so when we put, and I apologize, I'm going to make the blood vessels green so you can visualize them. So we're going to put our vasa recta in.
It's usually blue, right?
And they flow, this is going up the the vasa recta flow in the opposite direction. So not only do the vasa recta flow opposite to themselves, they flow opposite to the nephron loop. Okay? So we have another countercurrent flow. Our ascending and descending limbs of our nephron loop flow countercurrent to each other. The ascending and descending limbs of the vasa recta flow countercurrent to each other. And even these flow countercurrent to each other. Back to your comment, yes, that water being pulled out would dilute the particles that we have, except they move into the vasa recta. Same purpose, here's our collecting duct. Filtrate's going down, it's in parallel with the vasa recta. But again, as we add water channels and water is pulled out to concentrate our filtrate, the vasa recta pick up that excess fluid as well. So it helps to maintain. The vasa recta don't form the gradient, but they help to maintain it. So on this image here, you can see that out in the cortex, our osmolality is 300. And then in the pyramids formed by the nephron loops of the juxtamedullary nephrons, we have this gradient. Now it says 1200 here, maximum really is 1400. Our cortical nephrons have very short loops. They don't affect the osmolality because of that. All right. Now, somebody was talking to me about an animal in the desert that can really concentrate its urine. It almost produces a solid, uric acid is solid component crystals. The kangaroo rat can concentrate its urine to about 4,000 milliosmoles. What would you expect is different. If its blood fluid is still 300 milliosmoles, what different structural, anatomical structural difference would you expect if it can form an osmolality gradient of 4,000? Longer, well, both, but longer nephron loops. So the only way we can get higher than 1,400 would be to have a genetic mutation that made longer.
So proportionally, the kidneys are larger?
The proportion between, they don't have larger, huge kidneys because the whole rat itself is about the size of them. But proportional for the fluid volume and the material that's occurring. Now, we're not done with this imaging. So here is and in the morning class, I made it a mirror image of what I'd drawn on what was here on the screen. This time I made it the same so that you're not getting confused, flipping back and forth. So here is our descending limb. Here is our ascending limb and our collecting duct. Okay? Here's the vasa recta. Now, our descending limb is permeable to water. Our ascending limb pumps out the sodium, but is not permeable to water. And so we see that maximum difference of 200 milliosmoles between the interstitial or extracellular fluid and the descending limb. The ascending limb here. When we look at the collecting duct, notice that when this leaves, it's hypoosmotic. It goes into the collecting duct. Even though the surrounding interstitial fluid is at 1,200 milliosmoles, the filtrate is still reading 100. That's because your body has to produce, especially, not so much down here, but especially in this area, ADH, antidiuretic hormone, is required for the insertion of those water channels. So water channels are present all the time in the proximal convoluted tubule, all right, all the time in the descending limb, never in the ascending limb, and only in the collecting duct in this latter portion of the distal tubule under the influence of ADH. Now you have this image on the top of page 26. It's not that the flow keeps going into the filtrate goes into one nephron loop after another. It's showing a sequence of effects. which I'm going to show you through a series of slides because the way this is drawn is just difficult to visualize. All right, but I have a series of steps written below that. Again, before we look at those, here's the vasa recta in place. So our descending limb and ascending limb. And the flow is countercurrent to the nephron loop and parallel with the collecting duct. Okay? How our body does that with everything packed so tightly together that all the vasa recta by the collecting duct are parallel with it, at least physiologically, that's the way it works. Okay? Now, these series of images I formed after, it's just really frustrating to try to get this point across and the actual events that are occurring. And so this is from a different textbook that I used for a physiology only class about 15 years ago. But it was the imaging that came across was much clearer. So here we have, that's what you can do with paint and PowerPoint and stuff. So here we have a descending and ascending nephron loop that matches our pattern on the board. All right. We've done nothing to the filtrate except filter it and remove out the protein, the amino acids and glucose. So this would be what we would have at the distal end of our proximal convoluted tubule. In the medulla, the interstitial fluid is matching our filtrate and blood. So we're starting at kind of a level playing field, all right? And everything is 300 milliosmoles. So what side is impermeable to water? The ascending side. This is also the side that has pumps, okay? And so we're pumping out ions, and because it's impermeable to water, the water doesn't follow the ions. So let's turn on a pump at the bottom of the nephron loop. So we're moving sodium out, and it's going to go into the interstitial fluid, and so the filtrate becomes less concentrated, and the interstitial fluid becomes more. Because of the vasa recta, that doesn't become diluted out. Okay, the water is picked up, any other additional stuff is picked up, and now that higher osmotic pressure pulls water out of the descending limb, and that water goes into the vasa recta, and the filtrate in our descending limb becomes equal in concentration to the surrounding interstitial fluid. Okay, everybody okay with that? Because that's what's happening. You're just going to do that over and over and over and over.
We have to do it.
Oh, I don't mind saying it again if you're not.
So, but the interstitial fluid, that doesn't change because of the vasa recta? Is that the reason?
Well, it becomes more concentrated because of the salt. So we're moving salt from here to here. That doesn't go back. Now, if we pulled water out, it should go back to that. Because that additional water would dilute it. But it doesn't go back to that because this additional water is taken away by the vasa recta.
It doesn't change.
Okay? So now, if we do this all the way up, so this is our countercurrent flow, all right? We have our maximum, this would be our maximum gradient if we had two separate vessels flowing side by side. But because this side flows into this side, we add another dimension. And that other dimension is called the countercurrent multiplier. So it's not a countercurrent exchange. It is a countercurrent multiplier. That's just the nephron loop. So what we're going to do now is allow this filtrate that is more highly concentrated, because yeah, we've created a filtrate that's 400 instead of 300, but that means the maximum we can concentrate our urine is to 400. All all right? We're going to move this concentrated filtrate over to this side, push this up into the distal tubule, and bring in fresh 300 milliosmol filtrate from our proximal convoluted tubule. So that happens in the next slide. So now we've brought in fresh 300 milliosmol. We've moved some of our more concentrated filtrate to this side and moved some of our diluted filtrate up. Now when we turn on our pumps and we create that 200 milliosmol gradient again, there's our 200 milliosmol gradient as we moved ions out of the ascending into the interstitial space. Again, the osmotic pull will pull water out of the descending limb. And now, so this is always a maximum of 200 milliosmoles difference from the ascending limb, and always the same maximum difference in the descending limb. Hit the last diagram on your exam. Okay, and trying to figure out how to fill those numbers in. Now as we go up, we keep doing that. Notice what happens when we get to 200. We can't go from 200 to 300 and 600 because that would be too high. So this actually decreases a little bit. Some of those ions will be taken up by the vasa recta, but we're starting to see that gradient. All right, it's 400. This will then, if I complete it, it'll be 350. So we're starting to see a little bit of change. 350 to 500. Up here is 300. So 300 to 350 to 500. Notice what's happening to our filtrate. It's getting even more dilute as it moves into the distal tubule. Instead of 200, it's 150. So now we do that again. And that's how we get our range. So what happens is we can increase the overall amount of osmolality by moving the concentrated side over to this side and pumping out even more ions, using that number of ions to make this side even more concentrated, moving that over here, having even more ions to move out. And so when it first comes over, it's the highest concentration, because as it flows up the ascending limb, more and more ions are removed. So in our interstitial fluid of the medulla, that's why we see more ions at the turn of the loop than we do up here. Because as it flows up, ions continue to be moved out.
So descending uses the ascending to change to interstitial to change to descending.
It keeps going around. That's the multiplier effect. Okay? And we end up then with this result. Yes?
This could exist all the time in a kidney or kidney?
All the time.
Only when
All the time. Okay? The only time it would change would be if we had some problem with our pump. There are certain diuretics. What would a diuretic do? Would it cause you to pee more or pee less? Sorry, micturate. More. Okay? And various diuretics affect different parts of our kidney. The diuretic that affects this component is called a loop diuretic. It's affecting the loop. All right, so if a loop diuretic, how would a loop diuretic work? Would it add sodium potassium pumps to our ascending limb, or would it remove them? It would remove them. So if we have fewer pumps, then maybe instead of a maximum osmolality of 1400, we may only get a maximum osmolality of 800. So then when the filtrate passes out our collecting duct, instead of pulling out fluid from our filtrate to make a highly concentrated urine, we could only concentrate it half as much, and the rest of that fluid would be lost and we'd make more urine. Okay? So a loop diuretic would affect that sodium pump component. And we use diuretics when we're trying to decrease blood pressure. Maybe the congestive heart failure and it's not strong enough to pump a large volume. So we want to decrease the workload on the heart. Okay? So depending on the condition, we could affect ADH. There's potassium-sparing diuretics. We'll talk about that in a few moments when we get to the electrolyte function.
Can you explain again, why is it not a 200 gradient between like 850 and 1,000 and 850, 700? Why is it only 150?
This,
No, in the middle, interstitial.
from here to here. Not quite sure what your question
There
is.
and there.
Well, this is just a gradual change. Okay, the 200 grade limit is inside the cell, across the cell. So the cell that's forming this wall, its sodium potassium pumps would have to do a greater amount of work to keep the ions, the sodium ions, moving them against their gradient. So we're taking sodium ions from this fluid, moving it into the cell and out of the cell to a higher concentration. That's active transport. And that's the maximum amount of effort that those cells can do.
I don't, you meant like the
Right
1,000
now
to
it's even
850.
numbers and then it just.
I think it just matters that it's going down as it goes up.
Yeah, it's going to, this could go, I mean, obviously it's going to transition. This could be, you know, it's going to go, it's just going constantly lower. It's not that this drops from 850 to 700, it's going to go 850 to 845 to so on, okay? So, even though I have these numbers in the middle, they are reflective of the entire medulla. So, around the collecting duct is where that is important. So around the collecting duct, we have 300, 350, 400, 450, 500, and we get to our maximum amount where the tip of our nephron loop was. Okay? So when we come, we're going to take a break, and when we come back from break, we're going to look at how that then affects the movement of water out. But it's pretty basic. Okay? If we have no, and it is, if we have no water channels present in our collecting duct, then no matter how concentrated it is outside, it's going to be difficult for water to leave. So if we go back to this image, 20 slides later, so we go back to actually this image, even though out here it's 1200, without the water channels being present, we still have a very dilute filtrate. Okay? So now, when I talked about this morning, the only thing I had to drink was one of those chocolate drinks. About half the class had drunk at least 2 cups of water. I hadn't drunk anything in addition to that. So would I be more likely or less likely than the class to have aquaporins present in my collecting ducts? More likely. Because I would be more dehydrated.
Oh, right.
Therefore, my body would have a greater need. I'd have a higher amount of potassium and sodium ions in my brain. I would have a higher need to retain the water. So ADH would be released. Aquaporins would be inserted into these cells. And now the osmotic gradient would have the opportunity to pull water out of the filtrate. Now, if I had drunk, where's my water bottle? This is in my office and I hadn't used it, okay? So if I had drunk 5 of these since this morning, I would probably have made a few trips to the bathroom. I wouldn't need to reabsorb water, so I would have few to no aquaporins present. So rather than, one of the questions was, does this change all the time? So rather than constantly readjusting how concentrated this interstitial fluid is, the body maintains this gradient, and what changes are the presence or absence of aquaporins. Okay. All right, let's stop there and take a 15-minute break. And when we come back, we'll look more specifically at the distal tubule and the collecting duct for aldosterone and ADH effects.